Eukaryotic mechanism of DNA replication. Usually they are similar. The prokaryotic and eukaryotic mechanisms are similar, and this process is tightly controlled and it occurs at specific times during the cell cycle, as we said, and it requires that we have a template strand on which now we are making a copy. Then we have what we call primers. We also need raw materials such as nucleotides. We require enzymes and other proteins, and it's a process that requires ATP energy. There are three basic stages or steps of replication: initiation, elongation, and termination. So we are going to look at eukaryotic replication, and I have summarized it in this diagram. So this is our parental DNA. The dark blue is our parental DNA running from 5' to 3'. So these are parental DNA. For us to have replication, it must be opened up. Remember we said we want to copy, and it cannot be copied as a double helix. We need to get one parent strand and another newly synthesized strand, because we said that our replication model is semi-conservative. So the parental DNA has to be opened, and therefore the first thing is that we have proteins we call enzymes that we call helicases that unwind the parental double helix. They unwind. And this process occurs in different parts of the DNA because DNA is an extremely long molecule. So we have the helicases opening the double helix, and then to prevent the opened DNA from winding back, we have what we call single strand binding proteins that stabilize the unwound parent DNA. You can see them there in red. And then after that, now we have the enzyme that usually will now start synthesizing our DNA and adding nucleotides to create a new strand. And this is DNA polymerase delta. So usually it will now start making the leading strand that is synthesized continuously from 5' to 3' direction. So this is the synthesized leading strand, 5' to 3' direction. Now, on the other strand of the parental DNA that is 3' to 5', we also have a synthesis of a new strand, but now this one we call it the lagging strand, and it is synthesized discontinuously. And discontinuously is because we have what we call additional of RNA primers. You can see the red, the RNA primer is added by enzyme primase here. So this primer is made, and from the primer, then there is now the formation of, or the extension of addition of nucleotides by DNA polymerase delta. And once it is extended, usually it will form fragments because the primer starts, then addition of nucleotides, then another primer starts, addition of nucleotides, etc., etc. So it forms what we call Okazaki fragments. And these Okazaki fragments will need now to be joined so that we can have now a complete strand still running again from 5', so 5' this side towards 3' direction. So after the RNA primer is replaced by DNA, by now another enzyme. So this part of RNA primer is now going to be converted into a DNA. Remember, this is RNA, and we cannot have RNA in our final strand. So we are going to have this RNA primer being replaced by a DNA fragment by DNA polymerase epsilon. And then after it has been replaced, then the fragments are going to be joined by DNA ligase. Okay, so now it will join the DNA now into a complete strand, and all the Okazaki fragments will eventually be closed up. And now that is how we have DNA being replicated within the cell. And we are saying that this replication, what we call a replication fork, usually will occur in different sections of the DNA. And that is why now replication will end when replication forks meet. So we have different sections of DNA being multiplied because we cannot start from one end of DNA to the other end. It has to be in different sections, then they all meet. Once the replication forks have met, then replication comes to an end. We also have what we call termination signals that will also be issued, and DNA replication will come to an end. Another end is when we come to the end of the chromosomes, which are called telomeres. Now, just like any other copying that we can have, even as like if you are typing and adding certain letters, you're likely to make a mistake. Even DNA replication is likely to have mistakes as it is being copied. So we have what we call proofreading when there is an error, and that is done by the DNA polymerase delta. It has a capability of autocorrecting or proofreading backwards, such that if this is the strand that we are adding nucleotides. So the polymerase has added an incorrect nucleotide at this position. It will realize the mistake. So the mispaired nucleotide will then be proofread and removed by 3' to 5' proofreading mechanism. Then once the correct one has been added, then we can now continue adding the correct one. So usually we cannot have DNA polymerase unless there is an error or a mutation adding incorrect nucleotides without correcting. So it will add, if a wrong one is placed, then it has to go back, proofread, then correct, and then continue synthesizing in the direction of 5' to 3'. So that is the DNA replication in eukaryotes, and I have summarized both the different modes of replication that exist in living organisms and cells and the DNA replication in eukaryotes. Thank you. If there is any question, remember you can always text, you can always put at the description and comment. And again, if you have not liked this video, kindly like. If you haven't subscribed, kindly subscribe. Thank you.